My lecture today is an overview of nuclear medicine therapeutics and theranostic. To provide a background to dosimetry, lecture later. So, my lecture today will consist of discussing what nuclear medicine therapeutics and theranostics are, some current theranostic isotopes, some early history of nuclear medicine therapeutics and theranostics, examples of common theranostics currently, and a little word about the importance of dosimetry. So, the word theranostic actually is made out of two parts. Actually, it comes from two Greek words. The first word is therapeo, which means in Greek to treat. And that's how we get our English word therapy, to treat or to heal. And the second word is noxis, which means knowledge. So when you add the two Greek words together, essentially theranostic means to, to know it, to see it, and to treat it. Some versions, alternative versions of theranostic would suggest that you can say that it is imaging and therapy sharing the same target. Or a more general way, you can say precision molecular probes leading to targeted therapy. So actually, theranostic is not, well, there's some question whether is it just a fad or fashion. But if you look at the academic interest over the last 2 decades, you can see the publication is exponential growth. And in fact, this paper only stops at 2017. And if you look at the commercial interest in theranostics, you see that many traditional pharmaceutical companies have taken on a tremendous interest in research and commercial development of theranostics, with many large market players entering nuclear medicine arena, which is very unusual if you think about 30 years ago. And currently, billions of investment dollars and even more than 60 companies are developing some aspects of radiotheranostics or clinical trials in radiotheranostics. And some of the key players in radioligand or radiotheranostic therapy would include several names here, like for example, Novartis, Bayer, Telix. So really to really understand theranostic, we can start at a little bit of history. So in 1895, Roentgen, we know, discovered some rays that was able to pass through cardboard from a cathode ray tube, which was really unknown at that time. So he called it X, a variable X, X-rays. And he received the Nobel Prize in 1901. But at the same time, Becquerel in 1896, he noticed that there were some minerals that could blacken a covered photographic plate. And he therefore discovered that there were some radioactive rays, and he received the Nobel Prize, a physics prize in 1903. And subsequently, with that, a major milestone in nuclear medicine imaging and therapeutics was George D. Hevesy. He was the first person to use a radioactive isotope, lead-212, to track the translocation of materials in fava bean plants in 1923. And he started the concept of tracer principle. And with that, a very important milestone in terms of nuclear medicine therapeutics and theranostic was Dr. Saul Hertz, who discovered the use of radioiodine. In fact, it started when he asked the president of MIT whether iodine could be made radioactive. That was in 1936. And subsequently, with the cyclotron making the iodine, Hertz actually treated the first patient with a thyroid goiter with iodine 130 in 1941, and that started a milestone in nuclear medicine therapeutics. And in fact, it's very interesting. He did not use iodine 131, but he used a shorter half-life iodine isotope of iodine 130. And moving on from there, we know that isotopes has been used for a medical treatment. For example, iodine 131 for thyroid cancer, phosphorus 32 for polycythemia rubra vera, strontium 89 for bone pain palliation, samarium 153, yttrium 90, this is an antibody for follicular lymphoma, radium-223, yttrium-90 microsphere for liver cancer, iodine-131 lipoidol for liver cancer, rhenium-188 lipiodol for liver cancer, and yttrium-90 for radiosyinovectomy for rheumatoid arthritis. So the basic concept of radiotheranostics or theranostics is that you see it and you treat it. To see it, you have positron emitting isotopes like fluorine-18, gallium-68, carbon-11, or SPEC isotope that produces gamma rays like lutetium-177, together with beta rays. And then you have a therapeutic radionuclide which you can substitute, and such as over here, lutetium, iodine-131, iodine, yttrium-90, and actinium-225. So the interesting thing about unsealed radiation treatment is that a lot of it is based on radiation crossfire, and that introduces interesting concepts because unlike external beam radiotherapy, which is more focused and linear, unsealed sources radiation is actually more random. So that introduces interesting challenges, such as the need to, how do you quantitate the radiation dose to a tumor, for example. One very key concept in theranostic is the concept of radioconstruct. The idea of theranostic is that you have a radioconstruct with two poles. One pole, which is the chelator that holds the radioisotope, and the other pole is a ligand which targets the target that you want to target. So at the radioisotope pole, you have various therapeutic isotopes such as yttrium-90, iodine-131, samarium, as we said. These have different properties, such as different half-lives, different radiation types, different energies, different decay scheme, and even different chemical reactivities. On the other side, which is the ligand pole, you can have various constructs. You can actually have the radionuclide itself, but at the same time, you can also have a small molecule, or you can construct a peptide, a small chain of amino acid, or you can have antibodies, or some form of a nano construct, a nanoparticle, or even a microsphere. So if you are talking about the radioisotope pole, we know that from radiation isotopes that we have different types of radiation coming from isotopes. If we think very generally, we can get protons, for example, but unfortunately, we can't really use protons for therapy because proton emission is very rare, and it's only produced from very proton-rich nuclei, which are not naturally found, such as cobalt-53, lutetium-151. You can possibly use neutrons. So we do have a neutron capture therapy, which uses non-radioactive boron-10 to capture thermal neutrons, and then with that to release heavy particles for tumor high LET radiotherapy. A lot of times we generally use electrons or beta particle, such as from iodine-131, lutetium-177, yttrium-90, phosphor-32, strontium-89, and samarium-153. Of course, we get gamma ray, which is long-range electromagnetic wave with a low linear energy transfer, which is not really suitable for therapy, but it's good for the diagnostic part of theranostic. A lot of interest now falls on this internal conversion and Auger electrons. And some of the isotopes that do produce internal conversion and Auger electrons are iodine-125, indium-111, and currently now there's a lot of interest in terbium-161. In fact, if you look at the chart below, you see that there are some data that shows that the use of terbium-161 with internal conversion and Auger electrons show better absorb dose and higher cell killing as compared to lutetium-177. And then we do have positrons, which are antiparticle of the electrons, annihilates almost instantaneously to gamma photons. Can't really use it for therapy. And you have alpha particles, helium nuclei, which we now use for various theranostics, such as isotopes such as radium-223, actinium-225, astatine-211. On the ligand pole of the radio construct, you can try to modify it by various methods of radiochemical synthetic engineering in order to modify the target affinity and the binding to improve the binding affinity, and so allowing for improved pharmacokinetics and pharmacodynamics, such as the residence time, the volume of distribution, tracer excretion, and therefore to improve the delivery of these radio constructs to the target. For example, this paper, Nature, just about 2 years ago, they tried to covalently modify the radioligand so that it can bind to the target with a higher binding and a lower discharge rate. So now I'm going to move on to talk very briefly about 3 major theranostic in vogue today. The first one being the somatostatin receptor, which is, you will know that it is used to target neuroendocrine tumors, and this is known by the abbreviation PRRT. And the second one that I will just touch on is the PSMA, which is to target prostate cancer, and it's also called PSMA therapy. And the third one being FAP, or which I'll explain later, is a lot of it is used to target gastric cancer, pancreatic cancer, breast cancer. It's called FAPI therapy. So talking about the first theranostic, on this, the target that we are looking at is the somatostatin receptor. These are actually molecules on the cell membranes of various tissue. But very interestingly, the type 2 receptors are highly expressed, more than 95% on neuroendocrine tumors. And that makes it very suitable as a form of therapy targeted to these tumors. So the idea is to change the amino acid in this substance called somatostatin, because it's a receptor for somatostatin. You modify the analog of somatostatin, and as you can see on the table there, you can see by changing the amino acids of the sequence, you can increase the targeting to the type 2 receptors. And this is the idea of theranostic or PRRT on somatostatin receptors. In fact, when you actually use it to treat neuroendocrine tumors, it has quite a remarkable response. As you can see from the diagram there, patients with very high uptakes, foci of uptakes in the body, after 4 cycles of PRRT, you can see there's quite a significant reduction in the tumor, the avid spots. And there is a good overall response, of almost 80 to 81%, with a very good overall survival rate. And we actually did some dosimetry to look at the normal organs exposure, and they are reasonable and comparable to other studies. So that led to a phase 3, a very famous clinical trial called the NETTER-1 trial, where this radio theranostic was lutetium-177 doratate, was used to treat neuroendocrine tumors. And it was a multi-center study in 41 countries in 8 countries, 41 centers in 8 countries. And it showed a very remarkable improvement of an overall survival, as you can see in the KM Kaplan-Meier curve on the right. This was followed by a NETTER-2 trial, which they used the same therapy for a more advanced grade 2 to 3 neuroendocrine tumors. And again, it showed a good favorable response. So with this concept of theranostic, so people have thought about how do we improve this PRRT. And some people would say that you can actually combine lutetium PRT with yttrium-90 PRT. You can do it in alternate sequence. You can do radio theranostic with radio sensitizers to improve the response of the tumor. Or you could change the isotope to alpha particles such as actinium-225 or even terbium-161. Or you could use both alpha and beta particles in the same production. Or you could change the receptor antagonist, modify the ligand by adding some other chemical called EB, advanced blue, to it. Or you could inject the PRT, the radio tracer, directly to the tumor itself. So for all these things are very interesting, and they are all being tested. So the next theranostic that is in vogue currently is a theranostic on PSMA targets that is expressed on prostate cancer. So this is a molecule that is expressed on the membrane surface, and it's 100 to 1,000-fold times expressed in prostate cancer. And it tends to be expressed in prostate cancer with higher aggressiveness. And the imaging part of it is the PSMA ligand is labeled with gallium-68, which is a PET isotope, and it's used for imaging. And after you have imaged, you can actually label it with lutetium-177 and use it to treat the patient. So in this case, for example, the patient was treated you can see the metastasis has slowly decreased. And the one below is actually the actinium version of PSMA theranostics. Because actinium is an alpha emitter, it has a small emission of gamma rays. So the images are blur, but you can see roughly that there is some uptake in the lesions. So that itself has led to, again, a very big clinical trial, which then approved the use of a commercial production of lutetium-177 PSMA 617, and that production is now commercially available as a product called Pluvicto. And this clinical trial is called Vision, and it shows on the graph that there is a great significant response to this therapy compared to standard care. And there are many, many radiopharmaceutical trials for prostate cancer using this PSMA theranostics as listed here. The third theranostic on the current horizon is called FAPI. FAPI itself is a molecule, a glycoprotein that is on the cell membrane again, but it is associated with cancer-associated fibroblasts, which produces fibrosis in the tumor, so to speak. And you can actually engineer the ligands against this fibroblast activation protein. And one of the ligand is called FAPI-74, which is a pure diagnostic compound, which you can label with fluorine-18 or gallium-68, and it becomes the diagnostic pair of the theranostic. The remaining FAPI ligands can be labeled with gallium-68 or even and beta emitters and gamma and alpha emitters as well, and act as the therapeutic component of the theranostic pair. So when you label this gallium-68 to FAPI, you find that you can see on the picture here that quite a lot of tumors actually take up concentrate this gallium-68 FAPI, and it is particularly powerful in detecting metastasis from gastric cancer, from colon cancer, from pancreatic cancer, and from breast cancer. So that has led to a number of clinical trials with FAPI theranostics where you change the imaging isotope for therapeutic isotope, such as lutetium-177, yttrium-90, and samarium, and use it for therapy. So I'd just like to end off with two slides. This is a slide to show a very pure theranostic pair, which is iodine-124 and iodine-131. Iodine-124 is a positron emitter, and iodine-131 is a beta and gamma emitter. So in some thyroid cancer, they lose the ability to trap iodine, and therefore they do not respond to radioiodine treatment. But after certain chemotherapeutic drugs, some of these thyroid cells can redifferentiate to trap iodine, and some of these chemotherapeutic drugs are called tyrosine kinase inhibitors like dabrafenib and trametinib. So after giving these drugs, some of the tumor can actually take up iodine. So by doing iodine-124 PET-CT, you can actually determine which patient has responded to these drugs, and therefore the suitability. And by doing dosimetry, you can actually then use iodine-131 to treat these specific patients. And this is a very interesting, and in fact, also a highly topic that is being looked at currently. And finally, I just want to say that there's been much work on how to calculate absorbed doses from Y90 microspheres that correlate with response of the liver cancer. And part of the optimal use of radioisotope for theranostic is to understand how radiation affects different kinds of cellular tissues. And this is where radiobiology is very important, and the use of dosimetry becomes very important. And there is still much that we need to discover. And with that, I would just like to hand the time over to Prof. Rachel Chee to give us a talk on dosimetry. Thank you very much.